Hello and welcome to section 3 of our chapter 21 video lecture series. In this video, we'll talk about the adaptive immune response that involves our T lymphocytes and their functional types. So we'll start off with a little background. We are going to have our adaptive immune response involve the ability to recognize, respond to, and remember that particular substance. Whereas in our innate immune responses, we are ineffective at being able to control that pathogen growth because it's not specific to it. So we, using our adaptive immune response, will be able to tailor make an immune response so that we can have a strong, effective defense and be able to control or eliminate the pathogen. So we'll start off with primary disease and immunological memory. When we have that primary response to a pathogen, our B cells become activated by the antigen. Our B cell then proliferates, it produces a lot of plasma cells, which then create a tailor-made antibody to that pathogen, as well as what we call memory B cells. And again, that means the second time we are exposed to that same antigen, our memory B cells will proliferate and divide to form those same plasma cells and be able to generate the specific antibodies much faster so that we have a greater effective response. And you can see this in this figure here, where we have our primary response, where we were finally allowed at 3 to 14 days to be able to create those antibodies to combat the pathogen. Whereas that secondary immune response, when we come into contact with that same pathogen, we are able to form those antibodies within hours to a few days. And you could see a much greater response in response to that pathogen. So what are our stimulants for our T cells? They're going to be the antigens. And if you remember, those antigens are serving as IDs for our body. So when they are foreign, that means they're not produced within our body. They're coming from the outside. So this could be a microorganism like bacteria, viruses, a protozoan that can cause disease. But other times they can even be things that are harmless, such as pollen, animal dander, certain foods or drugs that can cause an overreaction of our immune system, which we call an allergic reaction. Now, all of our cells within our body have self-antigens, meaning they have these IDs that are produced by our body and recognized by our body. And so we use these markers to allow our adaptive immune response to be able to differentiate our own body cells from the foreign invaders. The response to our self-tumor antigens is helpful, and a response to our self-antigens can result in tissue destruction, which is known as our autoimmune diseases. Let's take a look at a typical T cell receptor that we're going to find on our T cells. This down here is our plasma membrane, and you could see that we've got this constant region that will always stay the same, and at the very tip here we have the variable region, and this is where we are going to change it based on genetic recombination so that we can attach to the different types of antigens that may be found within our body. Now on our antigens, we don't just stop there, but we actually have structures on here called antigenic determinants. These are going to be specific regions of our antigen that are recognized by our lymphocytes. So we can activate our lymphocytes in two ways. Using our antigenic receptors, so our T cell receptor that we saw in the previous slide, or our B cell receptors, which kind of look like an antibody attached to our B cell. And the other way is to activate using our glycoproteins known as MHC molecules. MHC stands for our major histocompatibility complex, and these are attached to our plasma membranes. and have a variable region, just as we saw on our T cell receptor, that can bind to our foreign cells as well as our self-antigens. Now let's take a look at the two classes of these MHC molecules. Class 1 is going to be found on our nucleated cells, so virtually all of our body cells. And of course, we have some cells that don't have a nucleus, such as our red blood cells, so they would not possess an MHC class 1 molecule. So pictured over here on the left, we have our target cell. This is just a regular body cell, and its MHC class 1 molecule has a foreign antigen on it. So our cytotoxic T cell was kind of roaming around and noticed this foreign molecule and took its T cell receptor and attached to it. So now this cytotoxic T cell is going to become activated, and it is then going to go through its process to eliminate the infected cell. Next, we have our class 2 molecule. This is found on the surfaces of our antigen-presenting cells. So remember, that's going to consist of our macrophages, pictured here, our B cells, and our dendritic cells. And of course, our macrophages are basically our monocytes just found within the tissues. So our MHC class 2 molecules here are going to display that antigen, and it is basically sounding off an alarm saying that we've got a foreign invader. So it stimulates our immune cells to then come and respond to the antigen. So in this case, our helper T cell is helping us do that. These next two slides are just showing you how we go through antigen processing and our MHC class 1 molecules. So again, this is just a normal nucleated cell. We have our protein here that is within our cytosol. We break it down, and within our rough ER and our Golgi apparatus, we are going to work with both of those organelles to create our MHC class 1 receptor and put that antigen or fragment on the receptor and display it on the cell surface. Now with our MHC class 2 molecules, we also have our foreign invader that we have taken up. We are going to put our MHC class 2 molecules with the antigen and then move it up to our cell surface so we can display it and trigger our immune cells to cause an immune response. Now, what about our T cell development and differentiation? Our immune system is able to create a tolerance, meaning we have a state of unresponsiveness of our lymphocytes to a specific antigen. Now, you might be thinking, well, don't we want our lymphocytes to respond? Yes, to our foreign invader, but we also need to make sure that we have unresponsiveness to our own body cells. So an unresponsiveness to our self-antigens. And so this tolerance can be provoked by a deletion of any lymphocytes that are reacting to our own cells, also by preventing activation of our lymphocytes that encounter self-antigens, and then activation of those regulatory T cells or suppressor T cells that are producing suppressive cytokines or kill antigen-presenting cells, again, attacking our own immune cells. But this is poorly understood at this moment. So next, let's talk through our mechanism of T cell mediated immune responses. We are going to have an antigen that our macrophage finds and realizes is foreign. It'll take it up and display it on its MHC class 2 molecule. And then our T cell receptor on our helper T cell is going to recognize it and bind to it. At this point, we have co-stimulation that's going to take place using the CD4 glycoprotein here. That comes from our helper T cell. Remember, we also call these our CD4 cells. And our macrophage is going to secrete a cytokine known as interleukin-1. That interleukin-1 attaches to our receptors here and stimulates the helper T cell to secrete another chemical known as interleukin-2. And what's interesting is that interleukin-2 will then go and bind to more helper T cells, and we will get a division or a production of more helper T cells, which we're going to call our daughter helper T cells. And the reason that we want more helper T cells is so we have more of an army to go over and activate our B cells to cause them to produce antibodies to also combat the pathogen. Or it can also activate more effector T cells. All right, let's talk about the CD4 and CD8 receptors. These are cell adhesion molecules that are keeping our T cell in close contact with the antigen presenting cell by directly binding to that MHC molecule. So we saw this in the previous slide, but you can see that combination taking place over here, and our antigen presenting cells are held together in two ways. It's by either this CD4 cell or the CD8 cell, which again will also attach to the MHC molecule. And in that way, we get full activation of either our helper cell for the CD4 or cytotoxic T cell for CD8 receptors, and they can trigger their response. So our helper T cells, like we said, that's going to release cytokines to kind of ring the alarm for our immune system, whereas our cytotoxic T cells will directly kill the infected cell by using granzymes and perforins. Perforins kind of form a whole similar to the MAC complex of our complement proteins, and the granzymes are the contents that move into the cell and will help lyse the cell. So this slide kind of reiterates what we said already about our helper T cell, and we newly are going to learn that we have two classes of helper T cells. We have our Th1 cells, which will secrete cytokines and regulate immunological activity and development. And our Th2 cells are also going to secrete cytokines, but these are going to act on our B cells to drive their differentiation into those plasma cells to then secrete antibodies. And our cytotoxic T cells, I mentioned, will work specifically against our virus-infected cells, our tumor cells, and any tissue transplants. So they work kind of similarly to our NK cells in the aspect that they both are targeting virus-infected and cancer cells. So we have this major lysin in perforin. We've talked about perforin before, in which we are going to form a pore within the infected cell, and that is going to allow a protein digesting enzyme known as a granzyme to move into the cell that will then trigger apoptosis intracellularly, meaning inside its own cell. We will also have our killer T cells be able to produce cytokines and promote phagocytosis and inflammation. So let's quickly talk through how we have the proliferation of our cytotoxic T cells. We mentioned how a target cell would have its class 1 molecule holding the antigen. Our cytotoxic T cell would then come over and the killer T cell will become activated. So we've got co-stimulation with its CD8 and other surface molecules here. And another co-stimulation is done by our helper T cell as it's secreting interleukin-2. So our T cell definitely is activated now and will divide into daughter cytotoxic T cells. And lastly, I've mentioned our regulatory T cells already. These are also known as our suppressor T cells. We don't understand them all too well, but they're going to have the CD4, CD25, and FOXP3 markers on them, and their job is to suppress other T cells in their response so that we can reduce the chance of damaging our own tissues and causing an autoimmune disease.